Yesterday we did the boil and cool, so now I have the tube with the 4 boiled and cooled oligos, but I need to set up a dilution, and as you calculated, I need to set up a 100-fold dilution. So what I'm going to do is get another sterile tube, and I'm going to label that with this particular oligo. Alright, so now what I need to do is take 100 microliters from my boiled and cooled oligos. I have a pipetter set for 1 microliter, always double check. Fresh tip. So I'm going to take 1 microliter of this boiled and cooled. It's not much volume. I'm going to shift it over and put it into my freshly labeled tube for dilution. So I have 1 microliter of dilute of the oligo from boil and cool, and so now I'm going to get water, 99 microliters of water, which is here, and take that. And now I always like to have the small volume in there and add the big volume to it, so it sort of mixes it up, but I can see that little tiny drop. So now I'm going to pipette up and down to mix it like that. Alright, so now I have my dilution of the boiled and cooled oligo. Above you can see the original boiled and cooled oligo pair, and below are the 100-fold dilutions of each oligo. And that's what I'm going to get ready for Golden Gate assembly, which I'm going to do here. So in these tubes are 9 microliters of the Golden Gate assembly mixture, and I have a tube labeled N for negative control, and so I'm going to take that, because that one's just water, I'm going to take water and put it into the negative, and I'm going to put my dilution and put it into the experimental, X for experimental. But again, I'm just going to take 1 microliter and add it to the 9. So here's my experimental. It's already well mixed. 1 microliter of the experimental. Move that over. Here's my pre-labeled tube. And I'm going to add that. Mix a little bit, make sure it's in there. Good. And now I'm going to take 1 microliter of the negative control, which is just water, for the Golden Gate assembly, because I don't want a promoter to go in there. Take 1 microliter of that, move it over, add it to the negative control tube. And I want to treat my negative control tube the same way I treated the experimental. So I flick it, do everything the same. The only difference is the experimental has the oligos, the negative control has water. So I'm going to set up the other 3, and then we'll go over there and do the Golden Gate assembly. In this microfuge rack, you can see at the far end are the original boiled and cooled oligo pairs. In the middle row are the dilutions, and the bottom are the Golden Gate assemblies ready to go.